Hello, good morning. Myself, I am BSR, botany lecturer. Today, our topic molecular basis of inheritance. Molecular level. What is the base for inheritance? Actually, nucleic acids are the basis of inheritance. DNA is the genetic material in majority of the organisms. RNA is the genetic material in plant viruses. Anyway, these are the nucleic acid, deoxyribose nucleic acid, ribose nucleic acid. So, before going to in depth, some introduction part we have to discuss. Especially when you take bacteriophage. In the bacteriophage, 5x174, 5x174 bacteriophage, how many nucleotides are present? Number of nucleotides. 5386 nucleotides. 5386 nucleotides means 2693 base pairs, nucleotide base pairs. Like that, when you go for bacteriophage lambda, number of nucleotide base pairs 48,502 base pairs. Like that, when you go for Escherichia coli, E. coli, how many nucleotide base pairs are present? 4.6 into 10 power 6 base pairs. In the haploid cell of the human being, haploid cell, there how many base pairs are present? 3.3 into 10 power 9 nucleotide base pairs. Whereas you can calculate in the diploid cell what is the length of the DNA. So length of the DNA in haploid 3.3 means in diploid 6.6 into 10 power 9 into 3.4 angstrom minutes is equal to 2.2 meters. So generally length of the DNA 2.2 meters in a human being. So it is the introduction. But we have to go in depth of the DNA structure, already discussed in biomolecules, but this chapter is different, that chapter is different. Again, I am giving clarity about DNA. Later on, we go for RNA. So DNA, it is the genetic material. Double helical model of DNA proposed by Watson and Crick. Double helical model proposed by Watson and Crick. And mainly Franklin and Wilkins, they conducted crystallography techniques. It means X-ray diffractions. Crystallography means X-ray diffractions. So they conducted the experiment crystallography. And this one DNA chemically analyzed by Chargaff. So Chargaff chemical analysis conducted by Chargaff, and there in between the nitrogen bases hydrogen bonds formation takes place, that explained by Paulings. So what Paulings proposed, purines and pyrimidines always exist in 1:1 ratio. Adenine always complementary with the thymine by 2 hydrogen bonds. Cytosine always complementary with the guanine by 3 hydrogen bonds. That is the introduction about the Paulings and Chargaff's chemical analysis, Franklin and Wilkins crystallography technique, double helical model proposed by Watson and Crick. According to Watson and Crick, the DNA made up of 2 strands. Each strand is anti-parallel and the 2 strands are look like a twisted ladder. It's a twisted ladder, just a twisted ladder. It look like a twisted ladder. Anti-parallel, it means one, it ends with the 5 and another one ends with third end. So 5 prime and 3 prime. Always the diameter of the DNA is about 20 angstrom units, remember that one. Length of the each coil is about 34 angstrom units. In each coil, 20 nucleotides are present, or 10 base pairs are present. 10 base pairs. Here, important thing, always adenine complementary with thymine by 2 hydrogen bonds, cytosine complementary with guanine by 3 hydrogen bonds. The distance between one nucleotide pair to another nucleotide pair is about 3.4 angstrom units. Like that, how many pairs are present? Already two are there: adenine, thymine, thymine, adenine, cytosine, guanine, guanine, cytosine, cytosine, guanine. 1, 2, 3, 4, 5, 6, 7. Adenine, thymine, thymine, adenine, thymine, adenine, like that. So, how many pairs are there? 10 pairs are present. The distance between one nucleotide pair to another nucleotide pair is 3.4 angstrom units. Like that, 10 pairs means 3.4 into 10 equal to 34 angstrom units, length of the coil. So, angle between one coil, one to another one, that is about 36 degrees angle. Anyway, these are the two strands. How ribose sugar, how nitrogen base, and how phosphate is present. So these are two strands. I am taking, this is the ribose sugar. Yes, this is ribose sugar. Look at here. So first, second, third, fourth. This is deoxyribose sugar, DNA strand. It means H and H, here H and OH, here only fifth carbon is present. Here, so this is the first carbon. So this is the ribose sugar. Ribose sugar is present. Here nitrogen bases. Suppose I am taking adenine. Here adenine is present. Two CN rings. Adenine means two CN rings. Here phosphate is attached to the fifth carbon. Like that I am taking second one ribose sugar. This is second ribose sugar. Here also H and OH. Here the fourth one. Here phosphate is fourth one. This is the fifth one. Phosphate is attached to the fifth carbon. So this is the third carbon. I am taking one more thing. Yes, you can understand very easily. So this one is H and H. Here H and OH. So first, second, third, fourth, and this one is fifth. Here also phosphate is adding. Here second carbon is having two H and H, but here third carbon is the functional group. It containing the first functional group. So anyway, fifth starts, third ends in one strand. I am drawing here only. This is the ribose sugar, and here phosphate, and this is the ribose sugar, and this is phosphate, and here ribose sugar, and it is phosphate. What they are telling is here only nitrogen base. What is the nitrogen base? Thymine. Thymine is, this is adenine, purine, this is pyrimidine. So adenine, thymine, their hydrogen bonds can be established. Suppose here cytosine is present. Cytosine only one CN ring. Cytosine is always complementary with the guanine by 3 hydrogen bonds. Like that here, suppose I am taking the thymine. Here the thymine complementary with the adenine by 2 hydrogen bonds. So phosphate is attached to the ribose sugar by ester bond. So suppose you take, this is the third carbon, third carbon. So third carbon and phosphate binds with phosphodiester bond. Like that, here also phosphate and third carbon binds with phosphodiester bond. Like that here also. So when you observe, this is one strand, this is one strand, this is another strand. Look at here, very simply. Polynucleotide. Nucleotide means ribose sugar, nitrogen base, and phosphate. Here also ribose sugar, nitrogen base, and phosphate. They are all always anti-parallel. Fifth starts, ends with third carbon. Here also it starts with the third carbon, ends with the fifth carbon. So 3' and 5' like this. So this is the DNA. So when you take the DNA, two functions are there, autocatalysis and heterocatalysis. Autocatalysis means always new DNA is forming from older DNA. That is called autocatalysis. In presence of DNA polymerase. But when you go to heterocatalysis, there two steps are there. Transcription and translation. Transcription I am taking. So here mRNA is forming, mRNA is forming from the older DNA. mRNA forming from the older DNA, old DNA strand. This process is called transcription in presence of RNA polymerase. RNA polymerase. So here DNA polymerase, there RNA polymerase. But here the specialty two strands act as template. Two strands act as a template. But there only one strand act as a template. Only one strand. So the formation of mRNA is a transcription. When the mRNA involved in protein synthesis, that is called translation. So that is the DNA. And I am going to directly RNAs also. All RNAs are formed from the DNA only. I am going very quick, fast. It is the introduction. Already we discussed in biomolecules. So mRNA, rRNA, and tRNA. Simply, I am going to complete. mRNA is a messenger RNA, rRNA ribosomal RNA, tRNA transfer RNA. mRNA single stranded, all are single stranded, but single stranded unfolded polynucleotide strand, no foldings. It is a straight RNA. This is mRNA. So this mRNA molecular weight is 5 lakhs Daltons, whereas it is 10 lakhs Daltons, whereas it is 25,000. It is 25,000 Daltons. Okay, here among all RNAs, the percentage of the mRNA is about 5 to 10%. Here it is about 75 to 80%. Here 15% among all RNAs. But when you go to structure point of view, mRNA is a single stranded polynucleotide strand. Single polynucleotide strand, unfolded, no foldings mRNA. In the mRNA, nucleotides are present in the form of triplet codons. The first codon is AUG, that is the initiator codon. Like that, total how many codons are possible on mRNA? 64 codons are possible. In that 64 codons, 61 are sensible codons. 61 codons code for 20 types of amino acids. 20 types of amino acids. What are the nonsense codons? When 61 are sensible, 3 are nonsense codons. What are they? UAA, UAG, and UGA are nonsense codons. So mRNA involved in protein synthesis. So mRNA is formed from the DNA. So DNA forms mRNA. mRNA involved in protein synthesis. So the amino acid sequence in the polypeptide chain. Amino acid sequence in the polypeptide chain depends on codons present on mRNA. So this is mRNA. rRNA, it is a ribosomal RNA. It is also single strand polynucleotide strand, but which shows helix. Foldings are present. So that is why due to the presence of pseudohelix is forms. A complementary with U, C complementary with G. By 2 hydrogen bonds, here 3 hydrogen bonds. but not exactly 1:1 ratio. Wherever helix is there only hydrogen bonds formation takes place. So among all RNAs, this percentage is more, that is about 75 to 80% present in ribosome. Then when you go for tRNA, it is the smallest RNA. This is also called soluble RNA or adapter RNA. Soluble RNA or adapter RNA, it is the smallest RNA. This tRNA clover leaf model proposed by Holy. Clover leaf model proposed by Holy. Especially when you observe in this clover leaf model, the one tail portion is present. That tail is ends with a third prime CCA end. That end is called cytosine adenine end. That CCA end is called amino acid binding site. Amino acid binding site. During the protein synthesis, amino acid attached to the third end. So this is the third carbon, this is the fifth end. Okay. Opposite to the tail portion, there is a loop which contain anticodons. So if there is AUG here, the anticodon is UAC. It is anticodon. So opposite to the tail, anticodon is present. On either side of the anticodon loop, one loop recognizes the ribosomes and another loop recognizes the enzymes. So the tRNA brings the amino acid to ribosomes for protein synthesis. So mRNA, rRNA and tRNA. Later on we will discuss about nucleosome.